Good morning, Javid. All right, today I'm going to go over the unit review for you. Okay, so we're going to go through this pretty quickly, but just look through this review in this PowerPoint, okay? All right, so first thing we need to know is the difference between sexual and asexual reproduction. Asexual is when one organism creates an identical organism. It's much faster and more efficient, but all the offspring are identical, all right? Sexual reproduction is when two parents contribute genetic material, and it's slower, but it's way more diverse and creates more variation in our world. All right, then you need to know the difference between phenotype and genotype. Phenotype starts with a P, is the physical trait. So it sounds like physical. And a great example would be like purple flowers, something you observe, okay? Genotype is the genetic makeup of that trait. So that'd be like the big A, the big A, or the little a that we had discussed when we talked about Punnett squares. You also need to know the difference between just definitions between homozygous and heterozygous. So homozygous, homo means same, so that's the same alleles for the trait. So that would be like a big A, or little a. I need to change that. That should be little a. Heterozygous is two different alleles. That's when they have a big A and a little a. All right, so homo means same. It could be two big A's or two little a's. Heterozygous is a big A and a little a. All right, just know about the cell cycle, just the generals of it. So G1, this right here, is when it grows. S phase is when the DNA duplicates or it goes from 2N to 4N. All right, and then G2 is when it grows some more. All right, just know that. This is a pretty big section here of the test. The mitosis versus meiosis. So mitosis happens in our body or also called our somatic cells. It leads to two identical daughters, and daughter cells are 2N, or they're called diploid. They have two sets of chromosomes, okay? Meiosis happens in our sex cells, also called our gametes. It leads to four different daughter cells, all right? So the four cells are different, and the cells are N, or haploid. They only have one set of chromosomes in there. And this is also where crossing over occurs. It's what leads to more variation. Right, so in mitosis, if a starfish lost one of its tails or one of its points, it can actually grow that part back. All right, but that would be mitosis doing that because that's a body cell. Meiosis, this is a good diagram for meiosis. So you start off with this cell here, and it leads to four identical cells that have half the number of chromosomes, but they're all different. Right, so there's four different cells that have half the number of chromosomes. Now this here's mitosis. You just need to know this. Be to identify these, okay? So if you're given this right here, you need to be identified. This is telophase. A good way to remember is T in telophase. 2 starts with a T. All right, this is metaphase. You can see all the chromosomes lined up in the middle. So metaphase M, middle M. This is just you have to know this is prophase. This is when everything's sort of just jumbled together, all right? And this is anaphase. And that means sort of like getting pulled away. So you can see the chromosomes being pulled apart from one another. So telophase 2, metaphase middle, anaphase apart, and then prophase is the other one. You need to be to put them in order. And the order, a good way to remember is PMAT, P-M-A-T, PMAT, all right? You need to know the Mendel's law of independent assortment. That just means that alleles separate during meiosis. And also it means that one trait is not attached to another trait. So just because a plant is tall does not mean it's going to have green seeds, right? We also need to know how to do Punnett squares. So this is what Punnett squares do. They show the probability of a trait being passed off to the offspring. It does not give an exactness of it or a preciseness, but it gives a probability of a trait being passed off. So remember we did this is what we did on the paper that one day. So remember to separate the two alleles and then put them either on the top or the side and allow the letters to meet. This is a good, click on this YouTube clip here and watch this clip, all right? Remember this is like the box we'll use, like this little box. You put the letters here, 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 and here, and then they meet in each box. But watch this YouTube clip here. That'll explain how to do Punnett squares to you again. Also need to know about Down syndrome. It's caused by non-disjunction. That's when chromosomes don't separate properly during meiosis, right? And you have a 21st set of chromosomes, specifically the 21st. This is called a karyotype, which is a picture of your chromosomes. And you can sort of see here for Down syndrome, it has 3 chromosomes right here. See, everything else has 2 chromosomes, all sets of 2. Right down here, there's 3 chromosomes there. This is what leads to Down syndrome, right? And that's everything in there. That's what your test will be about. So use this to prepare for your test, okay? Take your test today. Take your test today. It's very important. All right. See you later, man.